Oh, it's a video.
All right, let's get started.
Can you get a piece of paper?
Oh, it's really complex. You pull.
I just pulled it. Oh, harder. I didn't want to break it. Don't say anything to people. I knew you wouldn't, but I just thought because he had just said, and he got out of your class.
All right, you guys. Please be quiet. All right, so today is about enzymes. Enzymes are a type of protein.
Okay.
Oh my God.
Yes, they are a catalyst. That is their job. They are a catalyst. But if we're looking at what they're composed of, they are proteins. That means that they have an amino acid sequence. For my AP kids, you need to know then they have their tertiary and quaternary structure, right? And that's all based upon their amino acid sequence. So 8.3, I don't know that we need to know that. 8.4 is probably where we need to be looking at enzyme stuff, okay? Enzymes, the first thing that we need to talk about, okay, so we can say what they are and then we'll talk about some stuff that's important for them. So enzymes are proteins that act as a catalyst. Okay, and that means that they do what?
Speed up a reaction.
They speed up the reaction by lowering the activation energy. All right, so since that word's there, let's talk about what activation energy is. So activation energy is what?
Oh, how much energy is needed to get started?
Yes, how much energy is needed to get the reaction started? So not the best example, but since we've been studying it kind of recently, especially for my honors kids, an example of this would be glycolysis, right? We have to invest 2 ATP in order to make ATP, right? So that you can kind of use that as an analogy for activation energy. So it's the amount of energy that's needed to get the process started, but in the end, we're going to be making some stuff or releasing some energy at the end, right? So it's just an initial investment phase. And for my AP kids, this is the whole ball rolling down the slide, right? It takes energy to walk that ball up, but now it has all this potential energy, and then it can, once you push it, it can go on its own. All right, so graphically, the way that they represent activation energy, because you do have to be good with these graphs, right? They will potentially ask, you might potentially, they will, I might, AP might, ask you about this. So when they have it, they will have, they have it what looks like figure 8.12 in your book, right? So we have the reactants have this amount of energy. They need to have this amount of energy for the reaction to go through. And down here we have products. So these are the products. So this is when the reaction is done. These are the reactants, right? So we need this much energy in order for the reactants to actually turn into the products, right? So that's kind of how we graph the energy needed for the activation energy. And when we're graphing the what a catalyst does, it's going to decrease that. So instead of being way up here with energy, it's going to be maybe here with energy, okay? So because now we only need this much energy instead of that much energy, the reaction is going to happen faster.
The green line's catalyst.
Green line is catalyst. So this is with an enzyme. Blue is no enzyme. Okay? And this would be energy. Okay? And they have a picture of both on page 153. So 153, the black would be without the enzyme, the red would be with the enzyme. So any questions about activation energy or what the enzyme does just job-wise? No? Okay.
Wait, okay, when you're doing a graph, is like in an experiment, or is this gonna be an FRQ question?
This wouldn't be an FRQ question.
Okay. Anyway.
But this might be something where they show you this graph and say, what is the enzyme? Like, they could ask something like, what does the enzyme do? Like, what is this showing that the enzyme does? And you would have to say,
it would be the smaller line.
it would be the smaller line if they asked you to say which one it was, and if they ask you what does it do, you would say it decreases the activation energy.
Okay.
Do you know what I'm saying? So they can. Who knows how they're going to ask it? But they wouldn't ask this, I don't think, as a free response. Okay. All right. So that is the activation energy. I'm definitely not going to talk over you guys, so I can wait. The next thing we need to talk about is an enzyme shape, because and its structure. Okay, its shape/structure is important. If an enzyme structure is changed, then it does not function properly, right? So it has a very specific 3D structure. Okay. And what is one part of the structure that's really important?
Protein.
The whole thing is a protein. The activation site, or the active site. So a lot of times when they show enzymes, they just show them as kind of blobs, right? So we have a blob, but then we have some part of the blob that has a very specific shape. So let's make it this, okay? So it has this blobby protein that has this very specific shape, and that very specific shape is called the active site. Okay, so this part of it is called the active site. And that's really important because what happens at the active site?
You don't know
It binds.
Yeah, on your notes you draw how.
That's what the reactions bind.
The reactants, and what's the name for the reactants? We don't call them reactants, we call them substrates. So active site is where the substrate binds. Okay, so the substrate has to have a very specific shape, right? In this case, it would have to be something like that, okay? That would have to be the shape of our substrate. And they call this, the fact that this has a very specific shape that only fits here, a lock and key example, right? Only one substrate is going to fit into one enzyme, okay? An enzyme will only work on one substrate. That is it, okay? So if that substrate is, it takes maltose and it's breaking it into glucose, only one enzyme will do that. That's it, okay? That's all its job will be. Yeah.
What are the things that end in ACE?
ACEs are enzymes. So enzymes, all enzymes are going to end in ACE, right? Just like all sugars end in OS. All right, the last thing that I want to kind of talk about is a lot of times when it gives you examples and pictures and stuff like that of enzymes, it shows that that we have our substrate, right? And our substrate is going to come and bind with the active site. When it does, what happens to the enzyme when that binds? AP people. Changes shape and kind of, no. We have an induced fit, right? It will almost kind of lock it in, okay? But for the rest of everybody else, we have our substrate now in the enzyme. What is the enzyme usually going to do? It's going to create products, right? So these are our reactants. We need to have our products. Our products are usually going to be what? What do they do to the substrate usually? They usually break it, okay? They usually break it. So in this case, maybe we have a piece that looks like this and we have a piece that looks like that. But our products are usually going to be our broken substrate. So sometimes they'll just have something like this. Sometimes it'll be actual examples. Like maybe they'll say that this is maltose, and they'll ask you, well, what are the products then? And you guys would say? Glucose. Okay? Or it might say dipeptide, and you guys have to say amino acids. Okay? So sometimes it will just be general, but sometimes they'll give you specific stuff, and it goes back, touches back to, do you actually remember what those things are for when we learned about them? Okay? So be aware that there's multiple ways that you could be asked this information. Okay?
What was your pass rate on the AP?
73%.
Is that good?
The national average is 56, I think. Yeah.
Sugar always a carbohydrate.
Yes. That's the type of macromolecule it is. All right, so any questions about the shape or structure of an enzyme?
Okay, does the enzyme when the substrate binds to the enzyme changes shape?
To fit it. So obviously there's a bonding that happens along this, right?
What kind of bond?
Depends. Depends on the substrate. But there is bonding that happens.
So can we not
For yours, if you even remember induced fit, you're good. And then for you guys, we need to talk about inhibitors and stuff. They aren't as much. All right. Honors bio. You guys had to learn about inhibitors, right? Competitive and non-competitive inhibitors. Okay. So when we learned about that, inhibitors do what?
Change the shape.
My purple doesn't run as well as it used to.
Oh.
Man, I need a new purple. Alright, inhibitors do what? They inhibit. What does inhibit mean? They prevent the enzyme from doing its job, right? So inhibitors prevent enzymes from doing its job. Their job, yeah. And if they're preventing them from working, how do we usually measure whether or not an enzyme is working? By its what? By the reaction rate, right? So we measure whether or not it's working by the rate of reaction, or the reaction rate, right? Means the same thing. And we usually measure that by how many products are being formed, right? If we measure how many products are being formed, then we know how well the enzyme is working. Okay, that's its reaction rate. So inhibitors will do what to the reaction rate?
Slow down.
They're going to decrease it. Okay, so they're going to decrease the reaction rate. And inhibitors, we have two different kinds of inhibitors that you guys had to learn about. The first one is called a competitive inhibitor. Okay. A competitive inhibitor is something that binds where? It binds in the active site, right? Just like the substrate would. So what does that mean as far as it and the substrate? They have the same shape, right? They have to have the same shape. So it binds to the active site. So it has the same shape as the substrate. So if this is the substrate, I can spell, then the competitive inhibitor would have a very similar shape, okay? Similar enough that it is also able to bind into the active site. However, do we get any products made when it binds to the active site? No. So it basically tricks the enzyme, right? So the enzyme thinks that it's making something when really it's not, okay? So that is a competitive inhibitor, okay? The other one is called a what? non-competitive inhibitor?
What's it?
All right, the second one is a non-competitive inhibitor. And what does this one do? Where does it bind?
Anywhere else.
Anywhere else but the active site. Okay? Okay, when it binds to anywhere not the active site, what does it cause to happen?
It causes the active site to kind of like change so that the substrate can no longer.
Exactly. So just in case you didn't hear, when it binds to a place on the enzyme, not the active site, it causes the shape of the enzymes to change. Specifically in the active site. Okay? So once this has changed shape, it can no longer bind the substrate anymore. Okay? So now it no longer functions correctly. Okay? So one tricks it, competitive inhibitors trick the enzyme into thinking that it's actually working, whereas a non-competitive inhibitor is going to be something that binds, let's say it binds here. Okay, that's going to cause this shape to change here, and now this substrate no longer fits.
Is that like a triangle thing makes a site to change? Is that to change? It says under non-competitive inhibitor.
Says anywhere but not the active site.
No, go down one.
Causes shape change of active site to change.
Shape.
Causes shape of active site to change. All right, so any questions about inhibitors, what they do, the types of them? All right, so the last little thing that we need to talk about are what are things that affect, other than inhibitors, what are some things that will affect the reaction rate of an enzyme? Okay, so inhibitors will definitely affect the reaction rate. The other top two are pH and temp. Okay, pH and temperature are going to affect the reaction rate. Okay, temperature we'll do first. Okay, and remember there are graphs that go with these, right? So for temperature, we would have temperature on the x-axis and reaction rate on the y-axis. Okay, and most things are going to do best at about 35 degrees Celsius. But it could be whatever they give you, because it could be, here's a graph, tell me what is the best temperature, what is the best pH. And instead of best, what word do they usually use? Optimum, right? What is the optimum temperature? What is the optimum pH? And that's always going to be where the reaction rate peaks. Okay, so just so that you know, optimum equals a peak in the graph. Okay, because it's showing a high reaction rate. So a high reaction rate means that enzyme is working really well. If you guys are going to do that, then leave. Because I'm getting frustrated. Alright, so for temperature, okay, usually in our body, because we have a pretty consistent body temperature, it's about 35 degrees Celsius. Other organisms, it might be higher, it might be lower, okay? But how much reaction rate do we have at 0? We don't have hardly any reaction rate. Well, what do we have at 100? Not very much reaction rate. So for temperature, we have an optimum range. If the temperature gets too hot, what happens?
Denatures.
Proteins denature. Just think of Luke and his little joke. Okay? So too hot, proteins denature. What does denature mean? They fall apart. They lose their shape. Okay, so when they denature, they lose their shape. Okay, what if they get too cold?
So now you
They slow down too much. They do not denature. Okay, please do not tell me they denature at cold temperatures because they don't fall apart. If anything, they would solidify. Okay, cold temperatures are not going to make them fall apart, but it just makes them unable to function. Okay, they go too slow, just like our body does, right? If we get too cold, we don't function as well. Okay. All right, pH. Once again, we'll also have a graph like this, but instead of, we'll still have reaction rate on our Y, but instead we have a pH. Okay, now you guys can't get your pHs mixed up because they like to do this low-high. Okay, if it has a low pH, if it has a high pH, right? They like to do this to confuse you. A low pH would be what numbers? 1, 2, 3. This is what type of pH? Acidic. Okay, don't let that confuse you on a question. Okay, don't think low means, I don't know, whatever. 12, 13, 14 is going to be a high pH. This is going to be basic. Okay, because we are people, we are usually going to peak, our enzymes are going to peak around 7 because that is neutral. Okay, there might be some organisms that live in a very acidic environment, and so they peak in a low pH or something. And what's an example? Honors bio kids, hopefully. Or was it AP? I don't even remember. What is an example of an enzyme that works at a low pH? In our stomach. Right? And our stomach is low. What's a high? Our intestines. Right? So that was, I think, an example on one of the tests that you guys had to know.
Yeah.
So a stomach would peak at a high pH. Intestines would peak at a low pH. Or I flip what I just said. Sorry, stomach, low pH. Intestine, high pH. Okay? But most of the things in our body will be neutral. Okay? If it's too high, what happens? If the pH is too high, what happens to the protein? It will denature. It will lose its shape. If it's too low, it will denature and lose its shape. Okay, so the pH will actually make the protein lose shape. So for honors bio kids, that's all you need to know. Okay? So what are they? What do they do? What's activation energy? What are some things that affect the reaction rate of an enzyme? Okay? My AP bio kids. We have to do allosteric binding.
Oh,